Welcome back to the deep dive. So today we're tackling what really feels like one of the grand challenges in modern biology.
Oh, absolutely.
It's this question of how to systematically figure out what thousands of genes actually do.
And not just what they do in a simple sense, but how they shape a cell's entire identity, its full transcriptional profile.
Right. It's this classic problem in functional genomics that
Yeah.
always forces this brutal choice.
It's a total dilemma, yeah. On one hand, you've got throughput. You can do these big pooled CRISPR screens, test thousands of genes at once, which is super efficient.
But you get so little information back. You might just measure what if the cell lives or dies,
Hmm.
or if one protein marker goes up or down.
Exactly. It's low content.
Yeah.
And then on the other side, you have this incredibly rich, detailed content from something like single-cell RNA sequencing. You can see every transcript in the cell.
But then you're stuck doing it one experiment at a time in these arrayed formats. It's slow, it's expensive, it's not scalable.
It's the efficiency versus information density problem.
So the groundbreaking work we're diving into today, from Dixit and colleagues, describes a technology that was really designed to just shatter that trade-off.
Mm-hmm.
It's called Perturb-seq.
And it is a brilliant combination of the two worlds. It takes the scale of pooled CRISPR perturbations and merges it with the phenomenal richness of single-cell RNA-seq as the readout.
So our mission today is to really unpack this. We're going to get into the mechanics, the computational framework, and then the incredible biology it uncovered in immune cells.
And this isn't just a small step forward. I mean, the authors demonstrate the power of this right away by analyzing 200,000 cells in their very first experiments. That's a new paradigm.
That is high content at a scale that was just not possible before.
Mm-hmm.
Okay, so let's start with the engineering. How do they do it?
Right.
You have thousands of different genetic experiments all mixed together in one dish. How on earth do you track which gene knockout created which specific cellular response?
It sounds impossible, right? Like trying to find one specific domino in a massive collapsed chain.
Yeah, exactly.
But the secret is all in the delivery vehicle. It's the lentiviral vector they use. They engineered it to be its own tracking device.
Okay, so break that down for us.
It's really a four-step process. First is what they call pooled delivery. You just infect your big population of cells with a whole library, a pool of these lentiviral constructs. Each one has a single guide RNA, an scRNA, that's designed to target one specific gene.
And here's the magic, right? The identification system. How do they make sure that guide RNA stays linked to the cell it goes into?
That's step two, and it's the core innovation, the barcode bridge. The vector is built to do two things at once. It delivers the scRNA to knock out the gene.
The perturbation itself?
And at the same time, it expresses its own unique little name tag, a sequence they call the guide barcode, or GBC.
So the GBC is like a mailing label on the virus that says, this package contains the knockout for gene X.
Exactly. It's the physical address for that specific genetic instruction.
Okay, so now all the cells are in a dish, they've all been perturbed, and each one is making a little mRNA transcript of its own unique GBC. How do you read all that?
That brings us to step three, the readout. You perform single-cell RNA-seq on the entire pool of cells. And what scRNA-seq does is it captures all the mRNA molecules from inside one cell,
Including the GBC's mRNA.
including the GBC transcript, which they designed to be highly expressed so it's easy to find. And crucially, the sequencing process itself adds two more tags.
Right, the cell barcode and the UMI.
Yep. Every single molecule from one cell gets a unique cell barcode, the CBC, that's the cell's universal ID tag. And then every individual transcript molecule gets a unique molecular identifier, or UMI.
Let's pause on the UMI for a second because that's so critical for accuracy here.
Yeah.
Why is that UMI so important?
It's all about avoiding technical noise. When you prepare your RNA for sequencing, you have to amplify it with PCR, and that process can create, thousands of copies from a single original molecule.
Which would make it look like that gene is way more active than it actually is.
Precisely. It creates amplification bias. The UMI is a random tag that gets stuck onto the original RNA molecule before any of that amplification happens. So even if you end up sequencing 50 copies, they all have the same UMI, and the software just counts it as one.
So you're counting real molecules, not just PCR artifacts. It's quantitative.
It's absolutely essential for quantitative accuracy.
Okay, so that brings us to step 4, the final link.
The final link. So now you have your sequencing data, and because that cell barcode, the CBC, was put on all the mRNA from one cell.
The normal gene transcripts And the special guide barcode transcript.
You can just connect the dots. You use the CDC to group together a cell's entire expression profile, and then you look for which GBC transcript has that same CBC, and boom.
The cause is linked to the effect. The GBC tells you the perturbation, and the rest of the transcripts tell you the result.
At the single cell level for hundreds of thousands of cells at once.
And what's really cool is the flexibility. They can tune how many viruses infect each cell.
Yeah, by changing the multiplicity of infection, the MOI. If you use a low MOI, most cells only get one virus, one guide, so you're studying single gene knockouts.
But if you crank it up,
If you use a higher MOI, cells start getting multiple guides. And now you can start studying epistatic interactions, where the effect of knocking out gene A is totally different if you also knock out gene B.
you're moving from a list of parts to a circuit diagram.
Exactly. You're analyzing the wiring.
So just a quick technical check here. They had to make sure this all actually worked, right? That the GBC was detected and the gene was actually knocked down.
Oh, yeah. They spent a lot of time optimizing it. They got the GBC detection probability up to 94% in some experiments, which is fantastic. And they showed that, yes, the system works. In most cases, when a cell got a guide for gene X, the expression of gene X plummeted. The on-target knockdown was successful.
Okay, so first check-in. Perturb-seq is this incredibly scalable technology that bridges the gap between high-throughput screens and high-content analysis. It does it by using this elegant barcode system, the GBC links to the CBC, and it uses UMIs to make sure the data is clean and quantitative.
And you can tune it to look at single genes or complex genetic interactions. It's a really complete system.
But generating the data is only half the story. You now have this monster data set, 200,000 cells, each with thousands of gene measurements. How do you even begin to make sense of that?
That is the other half of the innovation. It's the computational engine they built called MIMOSCA.
MIMOSCA. It stands for multi-input, multi-output single-cell analysis.
Right. And its whole job is to take that massive, messy data matrix and turn it into a clean map of causal relationships.
So what's the model doing under the hood?
At its core, MIMOSCA uses a regularized linear model, specifically something called the elastic net. And what the model tries to do is predict the expression level of every single gene. That's the output, Y as a function of which guides are present in the cell, plus some other variables. Those are the inputs, X.
And the goal is to get this regulatory coefficient matrix, beta.
Exactly. That beta matrix is the prize. It tells you, for every perturbation, exactly how much it influences every single gene in the genome, and whether that influence is positive or negative.
So why a regularized model? Why the elastic net? Why not just a standard linear model?
That's a great question. The regularization is absolutely key here. In biology, you have this problem where you have way more variables than you have independent experiments. It's a high-dimensional problem.
Right.
Regularization does two critical things. First, it prevents the model from overfitting, from just memorizing the noise in the data instead of finding the real signal. It makes the results robust. And second, it encourages sparsity.
Sparsity, meaning it assumes most genes don't affect most other genes.
Exactly. It pushes the coefficients for the unimportant connections down towards zero. So what you're left with is a clean map that only highlights the strongest, most relevant regulatory links. It filters out the noise.
Okay, that makes sense. So it's about finding the true signal. But speaking of noise, single-cell data is famously messy. There's so much technical and biological variation. How does MIMOSCA handle that?
It tackles it head-on by modeling those sources of variation as covariates. So on the technical side, it accounts for cell quality.
Meaning, like, how many total transcripts you captured from that cell?
Precisely. If one cell just has fewer reads overall for technical reasons, the model knows not to misinterpret that as a biological effect. It factors that in.
Okay, that handles the technical side. But what about the biological success of the perturbation? Just because a cell got the barcode doesn't mean the CRISPR knockout actually worked.
Yeah. This is maybe the most clever part of the whole framework, the success rate adjustment. They use an iterative approach. It's kind of like the expectation maximization algorithm.
Okay, put that in plain English.
Think of it like a self-correcting thermostat. The model doesn't just blindly accept that the cell was perturbed. It has an expectation for what a successfully perturbed cell should look like.
For example, the target gene's expression should be near zero.
Right, so the model looks at the cell's actual expression profile. If it doesn't match the expectation, if the target gene is still highly expressed, the model iteratively downweights that cell's influence. It basically says, this one probably wasn't a successful knockout, and it learns to ignore it.
Wow. So the data itself helps to clean up the experiment. The cell's own profile tells you whether the perturbation was successful.
Exactly. And this iterative filtering process showed that, on average, over 66% of the cells that got a construct were actually functionally affected, which is a pretty great success rate.
So that handles failed experiments.
Hmm.
But cells are also just different from each other. They're in different phases of the cell cycle, or maybe they're slightly different subtypes. That's biological noise.
That's the final piece, adjusting for biological covariates. MIMOSCA identifies these underlying cell states and includes them in the model. And this was hugely important.
How important?
In the immune cell data, these pre-existing cell states explained up to 20% of the total variance in the data.
A fifth of the signal was just background biological state.
Yes. And if you don't account for that, you might wrongly attribute a change to your gene knockout when really it was just because that cell was, say, about to divide. By modeling these states, they can subtract out that background noise and isolate the true effect of the perturbation.
Okay, let's do our second check-in. MIMOSCA is this powerful computational framework. It uses a regularized model, the elastic net, to build a causal map and avoid overfitting.
And it meticulously cleans the data. It accounts for technical things like transcript counts.
It iteratively figures out which cells were actually perturbed, with over a 66% success rate.
And it models the underlying biological states, like the cell cycle, which can account for a huge chunk of the variation.
All of which makes the final circuit map really robust and trustworthy.
Precisely.
All right, so let's get to the biology, the payoff. They applied this whole system to 24 key transcription factors in mouse immune cells. Specifically, bone marrow-derived dendritic cells.
BMDCs, yeah. And they stimulated them with LPS, which is a component of bacterial cell walls. It triggers a really strong immune response.
So what did this massive, clean data set reveal about how these TFs run that response?
It didn't just give them a list of effects. It gave them the entire regulatory architecture. The model naturally clustered the 24 TFs into 4 distinct modules, M1 through M4.
And what do those modules do?
They controlled 5 different gene programs, P1 to P5. So, for example, the M1 module was dominated by STAT1 and STAT2, and its main job was to strongly activate the antiviral program, which was P1.
That makes sense. STAT1 and STAT2 are the classic antiviral response.
Totally. And then other modules like M2, which had TFs like SEB and RELA, and M4 with SPY1 and NFKB1, they were driving the main inflammatory responses.
But the whole point of a deep dive like this is to find the surprises, the things we didn't already know. What was the most unexpected finding about that M1 module?
This was fascinating. The model predicted that STAT1 and STAT2, these antiviral warriors, were also acting as strong repressors of the mitochondrial biogenesis program, P3.
Wait, hold on. They were shutting down the cell's power plants.
Yes.
In the middle of an emergency immune response. That seems completely counterintuitive. Why would a cell do that?
It's likely about resource allocation. An intense immune response is metabolically demanding. The cell has to pivot its entire economy. It has to stop investing in long-term infrastructure, like building new mitochondria, and redirect all of its energy and resources toward one thing: making defense molecules, like cytokines, as fast as possible.
So STAT1 is acting like a wartime general, diverting all resources to the front lines and shutting down civilian projects.
That's a great analogy. And this secret repressive role wasn't just a model prediction. It was actually supported by other studies in the literature that had shown STAT1 can inhibit mitochondrial biogenesis in other tissues. It was a known but underappreciated function that the model picked up immediately.
That's incredible. Okay, so what about the other modules? You mentioned M2 and M4 were driving inflammation.
This is where it gets really interesting, because they weren't just driving it together, they were in opposition. The model revealed they controlled mutually opposing cell fates.
What does that mean?
It means they weren't just turning genes on and off, they were actively pushing the entire population of cells toward different states. The M2 module pushed cells towards the mature, highly responsive inflammatory states.
Yeah, and M4.
M4 pushed them toward a different, mutually exclusive state, P2, which seemed to be associated with less differentiated, maybe even abortive cells.
So they're like two opposing political factions fighting for the soul of the cell population. If you knock out a TF from the M2 module,
the population balance shifts away from that mature inflammatory state. And if you knock out an M4 TF, it shifts the other way. They're antagonists, regulating the proportions of these pre-existing cell states.
and the model could even see the wiring behind this push-pull dynamic.
It could. It showed that the TFs within a module reinforce each other with positive feedback loops.
Team M2 forever.
Exactly. And at the same time, they actively inhibit the competing module with negative feedback. This creates a really stable, robust switch that controls this critical cell fate decision.
This is such a fine-grained level of detail. How could they be sure the model was right about a TF's specific role, like calling it a repressor versus an activator?
They could validate it against other data types, like CRISPR-seq, which shows you where TFs physically bind on the DNA.
And did they find a good match?
A fantastic one. For example, the model predicted that NFKB1, which makes the P50 protein, was acting as a repressor on the genes it was bound to.
That's weird because we usually think of NF-kappa B as an activator of inflammation. The classic activator is the P50-P65 complex.
Right. But what the model's prediction implied, and what the biology supports, is that during this specific LPS response, P50 is likely forming a P50-P50 homodimer.
A dimer with itself.
Yes. And that homodimer can bind to DNA, but it doesn't have the part that actually turns on transcription. So it just sits there, blocking the real activators from getting in. It acts as a repressor.
So Perturb-seq figured out this very specific molecular mechanism that P50 was acting as a repressor homodimer, just from looking at the downstream gene expression patterns.
That's the power of the high-content readout.
Okay, let's move into the final segment. You mentioned earlier that by using a higher MOI, you can study genetic interactions, or epistasis.
Yes, and this is where Perturb-seq really starts to explore the complexity of biological circuits. When you have two genes knocked out together, the effect isn't always just A plus B.
Right, you can have synergy, where the effect is way bigger, or antagonism, where they cancel each other out.
Yeah.
How did the MIMOSCA model handle this?
They just added interaction terms to the model. And from that, they could classify every pairwise interaction as either additive, synergistic, buffering, which is their term for antagonistic or dominant, where one gene's effect just completely overrides the other.
And did any patterns emerge? Were certain TFs prone to certain kinds of interactions?
They were. They found that combinations involving NFTB1, that P50 subunit again, were highly enriched for buffering interactions.
So P50 was often involved in dampening or reducing the effects of other TFs like STAT1 or RILA.
Exactly. And the most interesting molecular finding was that this non-additive regulation, this complex interaction, was most likely to happen on genes where both TFs were physically co-bound to the DNA.
So it suggests a physical requirement. To get this complex regulatory math, they have to be sitting on the same piece of DNA, probably interacting directly.
That's the implication. It's about forming these higher-order protein complexes on the chromatin.
And just to confirm this wasn't some weird artifact of immune cells, they also applied the whole method to a cancer cell line, K562, right?
Yep. They wanted to show it was a general-purpose tool. They targeted TFs, but also cell cycle regulators. And it really highlighted the power of the high-content readout.
How so?
Well, you could have two different gene knockouts, say for AURK and TOR1 AIP1, that cause the exact same physical phenotype. Under a microscope, they both just cause the cells to get stuck in mitosis. They fail to divide.
A low-content screen would just lump them together. Mitotic arrest phenotype.
Right. But Perturb-seq showed that underneath that identical phenotype were two completely distinct transcriptional profiles. The high-content readout could easily tell them apart and reveal their different underlying mechanisms.
Okay, so this brings us to the final and maybe most important question, scalability. Is this actually feasible for a normal lab? What's the guide to the miserly for running a Perturb-seq screen?
This is a critical analysis for the future of the field. They did a downsampling analysis to figure out how much data you actually need, and the answer depends on what question you're asking.
Okay, so what if I want to see the effect on one single individual gene?
If you need to nail down the regulation of one specific gene, you need pretty high coverage. They found you need hundreds of cells for each perturbation and about 1,000 transcripts per cell.
Which is expensive, a lot of sequencing.
That's the expensive version. But here's the real breakthrough. What if you only care about the effect on a whole program or a cell state?
Like the entire antiviral response signature, which is made of dozens of genes.
Exactly. They found that detecting changes in these broad signatures is way, way more robust. You only need a few tens of cells per perturbation, maybe 20 or 30, and you only need around 400 transcripts per cell.
Wow, okay, that is a massive reduction in the sequencing depth and therefore the cost.
It's a game changer. It means that doing these high-content, genome-wide screens that target cellular programs isn't just a theoretical idea anymore. It's now economically feasible with current technology.
So to recap, Perturb-seq, paired with MIMOSCA, is this complete, scalable framework. It gives you causal links between genotype and a really complex transcriptional phenotype.
The future for it is huge. You can imagine integrating it with other CRISPR tools like CRISPR interference or activation, or even using it for lineage tracing in vivo.
But the paper leaves us with a kind of philosophical challenge.
Yeah.
Even with this tool, the sheer number of possible genetic combinations, if you're looking at 3, 4, 5 genes at once, it grows exponentially. It's still an impossibly vast space to search.
Right. So to tackle that, we have to rely on a statistical assumption, the assumption of sparsity.
That most of those high-order combinations will probably have no interesting interaction.
We have to assume that. Otherwise, the problem is just computationally intractable.
So that's the provocative thought we want to leave everyone with today. The authors have given us the roadmap to scale this technology. So if it continues on this path, what's the one fundamental biological question that you could only answer by looking at the combined effect of, say, 5 simultaneous genetic perturbations?
Something previously completely unimaginable. Maybe it's multi-drug resistance in a tumor, or a critical decision point in embryonic development.
What's that one complex cellular secret you would want to unlock, now that you can finally track the result of 5 different targeted changes at once? It feels like this technology gives us the first real thread to start untangling that kind of causal web.